the next quarter of an hour or so, asking how that could possibly have come about. First of all, how's it done today in living cells? And it's a very complicated process. I can only describe it in a rather oversimplified form, and here it is. Along here, we have an RNA molecule with, I've shown them in triplets of bases. Those could be any one of the four, so you've got 4 of each, you've got 64 possible triplets along there. In some way or other, that is the code for a protein, which is up here. A protein is a string of amino acids, and you want to think of a protein as like a poppet necklace. You've got lots of little beads which fit together in what I learned as an engineer to call male and female fittings in order to produce a string. And you can fit them together in any order you like. Somehow or other, the sequence of these things along here, the bases along here, has got to determine the sequence in which the amino acids are formed along there. And it's done essentially, as I've shown it here, by a series of molecules called tRNA molecules, or transfer RNA molecules. They transfer the information from here to here, which have at one end of them exposed a little triplet. Our old friend complementary base pairing ensures that this pairs with that. This, needless to say, is called the anticodon, and that's called the codon. Attached at this end of each tRNA molecule is an amino acid. And depending upon what the triplet is down here, different amino acids are stuck on up there. And therefore, this tells which tRNAs to line up, and the tRNAs line up the amino acids. And then again, all you have to do is to zip together the poppets, and you've got a specific protein. And that's really all there is to molecular biology. You can, well, very nearly all. But there is a problem here, which I once referred to as the catch-22 of molecular biology, or of the origin of life. The catch-22, you will remember, is that if it, in Joseph Heller's novel, if you were in the American Air Force during the war, and you wanted to get out, as any sensible person would want to get out, because he was shot at by Germans. It was very dangerous and unpleasant. If you wanted to get out, the only grounds upon which you could get out of the Air Force was by proving that you were mad. But of course, if you wanted to get out, it proved that you weren't mad. So you couldn't get out. That's the catch-22. Well, here, the catch-22 of molecular biology is that you really can't have proteins unless you've got a lot of DNA to make them, to code for them. The messages down here, all the tRNA molecules and so on. But we've just seen from Leslie Orgel experiment that you can't have a lot of RNA accurately replicated unless you have some proteins to do it. So you're stuck. No proteins without RNA, no RNA without proteins. Well, we were stuck on this one for a surprisingly long time until it was discovered that, and I think it ought to have been discovered sooner, but it wasn't, that some RNA molecules can act as enzymes. Enzymes, catalysts, do not have to be proteins. And indeed, nowadays, you can hardly pick up a copy of Nature without finding an account of a new RNA molecule catalyzing a new chemical reaction of some kind or another. And this has led to the notion of an RNA world in which, instead of what we have at the moment, a division of labor between one kind of molecule which does the information carrying, the nucleic acids, and another class of molecule, the proteins, which do all the work, all the catalyzing chemical reactions and so on, connected by the code, which is here, you only had the one class, RNA molecules, and that's fine. But nevertheless, we've still got to understand how the code originated. And before I take this slide off, I want you to notice what the essential thing is. And it's indicated by that red arrow there. Somehow or other, you've got to attach a specific amino acid to a specific tRNA molecule, and a different amino acid to this tRNA molecule, and a different one to this one, and so on. You've got to make specific linkages between particular tRNA molecules and particular amino acids, and that's the nature of the code. How did that originate? And what we need is the notion of the fitting of a particular amino acid to a particular tRNA. At this stage, I want to talk about the ideas of my young friend Satmari, a Hungarian colleague, who has recently come up with an idea about the origin of the code, which seems to me, at least it's a jolly plausible guess as to what might have happened, and it's also one which, in the long run, I think is testable. Though at the moment, I think it still has to be seen as speculation rather than as obvious truth. It's based on, the basic notion is that if you come across something very complicated like the code, and you can't see how it could possibly come into existence because it's so complicated, the chances are that in the early stages it was doing something quite different. Perhaps a classic example, if you look at the primary feather of a bird's wing, perfectly shaped and formed for flight. But, poor old bird, if it wasn't perfect, it would... So how are we to explain it? Well, there's no question the first feathers in evolution were not to help birds fly. They were to keep them warm. They were down feathers. And anything that sort of splays out of scale a bit is going to help to keep you warm. And in fact, one can think of a series of intermediates between no feather and a perfect primary feather. But the important thing is that the first function of feathers was different from their final one. And if my friend Ersch is right, the first function of this linkage, that linkage there, in evolution, was nothing to do with the coding for proteins. His basic idea is the following. Here is a ribozyme, which is simply sort of gobbledygook speak for an enzyme which is made of RNA instead of a protein. It's an enzyme which catalyzes something. Now, many proteins have what are called cofactors. They have attached to their surface, at their active site, some compound which is not an amino acid, which helps them to carry out some particular chemical reaction. Perhaps the most familiar example is the way that the hemoglobin in our blood, hemoglobin being a protein, can combine reversibly with oxygen, and that's what its function is, to take up oxygen and release it again, and it does it with the help of an iron atom combined inside the heme element, which is a cofactor working with the hemoglobin. Ersch's idea is that ribozymes would have benefited very greatly if they could use amino acids as cofactors. Because amino acids are chemically more reactive on the whole than the nucleotides. What he imagines is that particular triplets of nucleotides, and there are reasons why it would have been a triplet, were attached to a particular amino acid. Different triplets being attached to different amino acids. And then, if this ribozyme wants that amino acid as a cofactor, it has to have a tag at the end here of 3 nucleotides with which that one can base pair, and then the cofactor will attach. It's just simply to improve enzyme catalysis by ribozymes. Nothing to do with coding for proteins at all. But if it's a good idea to have one amino acid, maybe it might be even better to have 2 of them. And if 2, why not 3? And you're beginning to see how this thing might evolve into a message, into the modern method of synthesizing protein. In a bit more detail, his notion is shown here. Here is a cofactor, an amino acid being attached to a trinucleotide by an enzyme. I'm not going to say synthetase, well I just did, but the reason I didn't want to is that I lisp, and if you lisp, synthetase is the sort of word that you try to avoid using. Anyway, there is a cofactor being made. It then binds to the ribozyme, which is simply replicated in the way that RNA is replicated. And he imagines this evolving into a situation where this ribozyme has become a message with a whole series of amino acids lined up against it. This thing here is another ribozyme. It's the analog of what today is the ribosome, namely the body where proteins are synthesized, and in particular where they're stuck together end to end. And it's interesting that the enzyme within the ribosome, which today forms the peptide bond, sticks the pockets together, is, as a matter of fact, an RNA enzyme and not a protein. It's a kind of fossil relic left in existing living organisms to give us a clue to how it all might have happened.